Okay, what we're going to do today, we will do is pound water for analyze APCs and coliform and generic E. coli. Okay, what is APCs? It is total aerobic plate counts. So which means how many total aerobic bacteria in the pond water. And usually people using APCs to do a standardization of the clean and hygiene, they have to lessen around 5 log 10 CFU per ml. Okay, it's just some standards used in the European country, but there's no definition, no defined answer for that. Okay, what's a coliform and generic E. coli for? Long time ago, people used this to indicate fecal contamination. So what is fecal contamination? Said very rudely is people or human beings poop to contaminate with the food supply chain. That's not good. You certainly don't want your vegetables have like a human stool samples there. Now, long time ago, about like 40, 50 years ago, people think coliform is a good indication, but later on the research said generic E. coli is a good one. Now, today, there's a little bit of difference between what we did before. Instead of we using an agar medium, we're going to use those paper medium. It's very nice, called a petrofilm. So all these products, we call it a 3M or petrofilm, because it's generated by a company, a big company called 3M. You know that during the pandemic, lots of the sanitizing hygiene gloves, like masks coming from 3M. So that's 3M petrofilm. It's a paper medium. I gave them a joke, I said it's a paper medium, which means all the nutrition ingredients is in this dehydrated paper. So when you see here, 3M AC, which means aerobic plate counts. And we have another one. If you could see here, I need to have a scissor to open this. Do you have a scissor? Okay. We need to have a scissor to open this. This is for E. coli. E. coli looks a little bit different. Looks like that. This is for E. coli and coliform, and this is for the APC. Okay. Now, we also have to do a serial dilution. So when we do it, we have to guess how many coliform and APC in the pound of water. So you get a bottle of pound of water, let's say, a bottle of pound of water. You have to shake the first thing, okay? You usually do shake 30 seconds. Mix it. Now we have to do dilution. What we do, you're going to get 2 tubes. 2 tubes, both are 9.9 ml, 0.1% nutrient broth. It's a buffer water. So we get 2 tubes. Since it's 9.9 ml, so what we're going to do is so-called 100-fold serial dilution. So we're going to be adding, using pipettes, 0.1 ml onto the first tube, mix very well, shake, and then you add another 0.1 ml onto the second tube. Okay, now here is a tricky thing. It's a little bit different from what we did for our agar plating. Because agar plating we did is 0.1 ml onto the agar plates. For the petrofilm we have to add 1 ml. Therefore, the tube dilution and the petrofilm dilution are the same, or exactly the same. Okay? Therefore, what's the dilution factor of the first one? Is 10 to the 2. And the tuber dilution is also 10 to the 2. What's the tuber dilution of the second one? Is 10 to the 4. And the final dilution is also 10 to the 4. And we're going to do both of these two for APCs and coliform generic E. coli. Okay, here is something I want to mention because we don't have enough material. So for APCs, everybody got 2 pieces. 10 to the 2, 10 to the 4th. For the coliform, 2 of you got 2 pieces, which means everybody got 1, and you work with your bench partner, you do 10 to the 2nd, your bench partner do 10 to the 4th. If you're by yourself, you can do it, all of them, okay? Because this is much more expensive. And if you look at that, this bag is only 25, and this bag is 50. Okay, I don't have them much, so a little bit difference, okay? So APCs is 2 pieces, 1 student. And the coliform and the E. coli is 1 piece, 1 student. Okay, you guys work together. And all these 2, you need to be diluted at 1 ml pipettes. Okay, so this is going to add 1 ml, which means it is 1,000 microliter. Okay, that's 1,000 microliter. And for the APCs, we need a presser. I will get a presser real quick into my lab. So you guys is that get the plates, get the petrofilm to do the label first, and do the dilution first, and wait for me to have the presser. And I will show you real quick how to do. And for this petrofilm, you just be gently cover it. It's good. But I will show you because you have to avoid the bubble. Now the next question is, how do we know it is APC? How do we know it's coliform? How do we know it's E. coli? It's a little tricky, so we want to talk about that. Okay, in this yellow APC petrofilm, after incubation, 35 degrees Celsius, 24 hours, sometimes we have to do 48 hours, okay? You see these very tiny dots or pink colony. We call it APC, aerobic plate counts. Now the tricky thing right here is this petrofilm. This E. coli coliform petrofilm is a little tricky. What's a little tricky about that? If you see those tiny red dots, those are coliform. And then if you see those red dots, a little bit purple, and they have like a bubble there. So we say blue to a purple colony with gas bubble. That is E. coli. That is generic E. coli. Generic E. coli. Now what is a total coliform? You have to add up them together. This is called total coliform. Okay? But sometimes you need to know, if it's really dominated by fecal contamination, you may find all of them are E. coli or generic E. coli. And sometimes, if it's not fecal contamination, it's just some of the soil dust or debris, you may find all of them are coliform. So it depends, but the previous experience told us the river or pond water near WVU campus have both. At least you will see generic E. coli. That means some of the poop samples are there. I don't know where that comes from. It could be close to the farm, could be close to the hospital. Could be somebody just dumped there. I mean, it happens. So last couple of years, we all find at least there is a couple of sample from 32 students. You have like 3 or 4 which is have generic E. coli there. That's not looking good. Okay, so that's a procedure. So what are you going to do? Realm will help you, everybody, to get 2 pieces of APC petrofilm, 1 piece of generic E. coli, coliform petrofilm, and start to label, started to do dilution. And this dilution is still use 0.1 ml pipettes, but finally you have to add 1 ml onto the petrofilm. And then I'll get a pressure and I get a fact sheets, and we're good to go. Okay? Very good.